So first, this is me. I'm Jesper. I will say a few words about myself and what we're doing. Essentially, we're based in Stockholm here in Karolinska, and I'd like to begin with this slide. In one sense, all of you traveled from different parts of the world and you're here. What I mean with this slide is not only this. In one sense, all of us are travelers, especially when we deal with systems like in systems medicine. We come from different backgrounds, we have different training, we have different mindset and different languages in one area moving into others. So we travel in an intellectual landscape. So I think you will meet fellows here who have different kinds of thinking than you have. And also in your research, you will meet colleagues and fellows with that kind of sort of background. And I think this is fairly unique for this kind of field, and that makes it also very exciting. So we're going to have clinicians talking with molecular biology people and mathematicians talking with physics people and so forth. So we are all travelers in this sense. So with that said, I'm going to say a few words about myself. Okay, this is me. So I have a diverse background. We're studying math and physics and also medicine and also some degree of social sciences like logic, theory, knowledge and these kind of things, because when you think about subjects, everything is really connected. Then I did a PhD in medicine doing experimental work and also computational work. Spent some time in Boston finding a sort of hotspot in the world where you have these kind of centers, multidisciplinary centers, that was really, really useful, and especially to meet people there. And since then, coming back to Sweden, me and my team have been doing this kind of work. And then I'd like to show you some slides on what we are doing, the kind of thinking behind it, basically, which I think provides an example. And we welcome you to visit our homepage. So, in one sense, we're trying to do three things, three perspectives here. One is, of course, for the benefit of patients. We want to understand diseases. What is wellness? What is disease? What is causing diseases? How can we prevent disease? That's a sort of core mission. And we like to do that by really understanding the molecular circuitry in cells, between cells and tissues. What happens when these things go wrong? And the third leg is really, for doing this, we need these kind of computational techniques to understand these kind of systems. And eventually we may be able to learn something from lifelike system, and we can come up with smart new technologies also. So these are sort of three legs that guides our thinking in this. And we're interested in health and disease, trying to understand networks of different cells, also how individual cells are working, but while doing this and trying to understand fundamental questions on what are cells really and how do they work, because it's in one sense a mystery still. And out of this we can sort of extract technologies and useful tools for actually understanding these systems. And in practice that means that we as a group, we publish like many systems medicine and systems biology groups in sort of what can look at the surface a bit different areas, but in one sense things come together when you travel in this way. So in practice in our team it means that we are around 25 people with very different training, different walks in life, and I think that is a characteristic of many of the research group you will encounter in systems medicine and systems biology, taking these kind of systems approaches to really understand diseases and developing new tools for understanding diseases. So I think we are a very sort of typical group in that sense. So this makes this endeavor really exciting, I would say. Okay, so much about us and our perspective. So the summer school, we're here, we're meeting here. It's sort of supported by Casym and FEBS. So Casym is coordinating support action and really trying to work out how should we sort of pursue systems medicine at the European and international level basis. So it's a consortium of research group around the world, mostly in Europe basically, and it's a very open sort of federation in that sense, and David will talk more about these kind of things. So what was the idea with the summer school here? So on the one hand, we develop a roadmap what kind of problems are important, what are the challenges, and how should we pursue them. But we also like to sort of develop a summer school starting like a tradition, and we had really two kind of ideas in here. The first one is that since we and others are travelers, there are many different perspectives here. So one idea is to invite researchers who are doing these kind of approaches to understand diseases and developing tools and show you examples of success stories, but also showing you examples of what are the challenges here, what are the kind of problems these groups are dealing with basically. So it's like a Swedish term, a smorgasbord of different things here, different perspectives. So that's, I think, what we can accomplish in 1 week, to have a taste of that. So that is one very important goal. And you should feel selected here, because the idea here is to attract people to this summer school who will become, and who already some of you are already doing this kind of work and will become leaders in this area here, setting the stage. So that's the first objective. The second one is really to initiate an annual meeting place where faculty, students, and others meet here on a regular basis at different places in Europe to discuss research challenges and come up with new ideas, new collaborations, to have that as a tradition basically. And this idea mimics to some degree ideas of schools like in Woods Hole and Cold Spring Harbor, for example, in biology. So that's the kind of ambition and thinking behind this. And we are here, you're in Esset. It's a very nice scenery. Today we don't have that much sun, but I'm told that there's going to be more sun in the week, apart from the rain tomorrow, but otherwise it's going to be fine. So this is a really beautiful place out in the archipelago in Stockholm, so that would be a nice sort of starting point. Now, at this point, you should be a bit confused. I've been talking here now a couple of slides, but what am I really talking about? What is this systems medicine? I did not sort of mention that really, what it is. So you should think about this, or in a more modern version, what do we mean by this? So and then we'll show one slide on that essentially. So this is a text extract from this roadmap, which you can find on the Casym webpage. So let's have a look at that text basically. So we can begin at the end here. The outcome, whatever we do in this box, it's supposed to make an improvement of health basically. That's the sort of target basically. We're going to make a difference in understanding and also in practice using these kind of systems approaches. And that should be a tangible kind of goal, and it should be measurable also. And then how do we do that? Well, that's, as I said earlier, a combination of mindsets and tools, computational part, statistical mathematics, and clinical perspectives, molecular perspectives. So combination. So one can sort of argue, should one try to define an area or not? That has been a discussion. And I don't want to hold David responsible for this formulation, but I'm just showing your image here because you will discuss this much more than I will. What is systems medicine and what do we mean by that? Okay, so much for the introduction on the concept of summer school and systems medicine. So let's move to the historical and conceptual perspective here. Where did we come from? Systems medicine. So let's look at the medicine part. Medicine leading into diseases and what I will refer to anomalies. I mean, observations that we have problem to fit into this scheme basically. So historically, medicine is a very practical thing basically. You feel, you touch, and you still do, and you try to understand what's wrong basically, and you give some advice. So this is really early on, back to the Greeks basically. Then later, when science is progressing during the centuries, we sort of build up a rich body of knowledge, lots of different textbooks in medicine. We start to begin to understand the anatomy of the body, and then we can give a bit more rational advice on what to do when something is wrong, basically. And this is, to some degree, how medicine is still practiced. We feel, we touch, and we observe, and we have some kind of measurements that can sort of facilitate that understanding. And basically, we try to understand diseases in terms of the kind of symptoms they show. That's the kind of the core idea. Now, it has become a bit more problematic, this medicine disease part. As you know, most of you are well aware of the sort of success of the molecular revolution, basically, with the really initiation of the Human Genome Project to really characterize the human genome. At the turn of the century, it was announced to be read for the first time, basically. But also, very importantly, after this revolution of being able to read the DNA, the last 10 years, lots of new technologies has been developed in order to measure aspects of the molecular machinery. And that's really fantastic. That's a revolution, truly. And of course, how we measure things and how we see things, that's also how we think about, for example, in this case, diseases. But we also know historically that whenever we have a new technology, revolutions are poised to occur. So for example, Galileo, at that time in physics, when people really started to being able to build telescopes, meaning that we can look and study things beyond what we can see with our eyes, and we can make measurements and detailed recordings, and then we can start to think about those measurements. That is really sort of changing the perspective from the Earth being the center in the universe to that, well, the Earth is moving around the Sun and the Sun is doing something else. So that changes perspective, and that's something being discussed very much in this very famous book by Thomas Kuhn. He's a philosopher of science. I think that was written in the 60s. So what drives scientific progression? You have normal science in one sense. We understand things, we add on knowledge, but at some point, some kind of observations come that we cannot fit into our current conceptual scheme of understanding something, anomalies. So when dealing with that, often a revolution happens, a paradigm shift. We see things in a different ways. And if we think back here, going from sensing and feeling and trying to understand diseases, when we have all this kind of molecular information at different scales, in space and also in time, we have many data points and measurements which is difficult to fit together in a sort of whole, basically. So I would argue that with these new technologies, like the telescope, we are on the verge now of sort of redefining what we mean by diseases and the human body and how cells are working. So we are struggling to come up with sort of a new perspective, new concepts on how to deal with this. Personally, I would like to argue that this is very important, because in many cases we think that limitations is how can we store the data, what kind of technology do we have to measure different things. That's very important. But very important is, of course, what kind of concepts do we have to understand these kind of systems. And I think we need to be more innovative and trying to really think hard and in practice trying to understand data and these systems, coming up with new concepts and ideas. So I think we are right now at this point in time in the middle of a revolution, basically, that 30, 40 years ahead in time will become very clear. So that's the medicine part. Technologies are driving us into a point where we have difficulty in understanding these complex systems. So this is really an exciting time, therefore. So let's look at the idea of systems, the other part of systems medicine, a bit historical and also conceptual. Systems, how that play into life, basically, and the idea of using mathematics. So what is a system? Some of you have a training in biology and medicine, and some of you have a computational training. And eventually, most of these things lead to sort of mathematical kind of formulation. So what is a system? In one sense, it's very simple. A system is really two things. You have entities. You have a graph, essentially. You have nodes, entities, things. These could be cells. These could be genes. These could be organs. You have things, and things which are connected. They interact somehow. So then in essentially abstract way, you have a graph. You can think about this in graph theory. But you don't only have this structure, you also have a dynamics. This graph is changing and moving, and things are happening over time. And this is something we can capture in mathematics, exemplified here by differential equations. So you have the nodes, z here, for example, that changes over time as a function of other things in the system, of other nodes and some parameters describing the interactions and dynamics in this system here. So in this sense, it's sort of a well-defined object. Well, what will happen in this system? Well, it will depend on what's the current state, what kind of input comes into the system, how it's hooked up, basically, what is the sort of time dynamics in the system, and it will evolve over time. And of course, connectivity, how things are connected to each other, is of course very, very central. That's a central property of this system. So this is a simple picture. Now, how can we understand this? So we know, for example, from physics, if you take a pen, you throw it up, it will fall in a specific shape, usually an elliptic shape. Wherever you do it, in South America, in Stockholm, or elsewhere, you have this kind of shape of objects falling under gravity. Planets are moving, not in a circle, but like an ellipse. You push the circle and you have this kind of movement. That comes up again and again. The same kind of dynamics in many, many different kinds of systems. So a pen is different from some other object, but they still follow the same law. So then it becomes a bit more complicated. How can we discover the essential laws and features in this kind of system? So it's not that simple, just it's a matter of formulating the equations and then study the complete system with all the details. It will not tell us everything, basically. We will have difficulty in discovering what are the kind of laws in here. What are the laws in biology, basically? Can we discover any kind of laws, basically? So this leads us into the analogy with physics, because we know from history that mathematics has been sort of very, very central in understanding these systems. So mathematics must be important in understanding these kind of connectivity and how the dynamics evolve. So this is most likely an approach that is useful for studying lifelike systems, which is a bit different from the sort of physical equations. But then if we take a further step back and look at some of the giants in physics. So the revolution in 20th century of physics, taking part the first 50 years, when many things in physics were formulated, the quantum mechanics, really understanding particles. So the reasoning here around the 30s, many physics people, 30s, 40s, and 50s, and also 60s, became very interested in lifelike systems like in biology. What are those? Because we don't see life when we look at Schrödinger's equations, for example. We don't see any cells in there. They describe very fundamental objects, but we don't see it there. So this is an example of, he's writing here about the problems in physics, nature of atomic nuclei, to be followed by more difficult ones, such as the problem of life, the solution of which problems will presumably require a more drastic revision of our fundamental concepts than any that have gone before. So he kind of anticipates that to understand these kind of systems, we most likely need other additional concepts to the ones we have been using in physics here. Quite likely these changes will be so great that it will be beyond the power of human intelligence to get the necessary new ideas. by direct attempts to formulate experimental data in mathematical terms. So maybe it's not enough just to capture everything in terms of equations. We need a way to understand these systems. And you know that you have a tension in physics between small parts and a big scale, how to put things together. And that's exactly what we also have in biology, I would say. So that was what one giant began to think about life, basically, the problem of life. And then it's a very famous book that most of you have heard of. And if you haven't heard about it, I would recommend you to read this book. It's written in the 40s based on some lectures Schrödinger, one of the giants in physics, wrote. So he and others started to really think about this. So what is the problem of life? Essentially, it comes in two parts in his mind at this point of time. One is, how do we transfer genetic information? How can we inherit properties from one generation to the next? There must be some kind of structure that mediates these kind of properties from generation to generation. He was thinking about crystals and periodic kind of structures. And this was really at a time before the discovery of the double helix in the 1950s, basically. So there was lots of interaction between physics and biology, seminars, workshops, and really traveling across boundaries here. And the second problem, as he saw it, in addition to how do you pass properties from one generation to another, was the issue of stability and order. How can these systems, and systems, he had this kind of picture in mind. You have elementary particles. In his days, you talk about the atom, protons and neutrons and electrons. How could these sort of large systems of these things connected in a very special way, how could they support and sustain order and structure over time? That is kind of a mystery if you think about it. So that was the second kind of conundrum that he was sort of thinking about. How is this possible? What kind of laws are sort of making these systems, which look like they are self-organized, to have a structure that persists over time? Eventually we will die, of course, but biological systems persist over time. How is this possible? Why don't just the atoms go in different directions and nothing will ever kind of survive in a stable structure over time? That was the second kind of question he was sort of discussing and thinking about here. So taking one example, what is a system now? A composition of atoms, yes, but let's look at the heart, for example. The heart viewed as like in a classical physiology book. So the heart is very simple. What does it do? It just pumps blood, basically. This kind of pump. That's a very simple system. Blood coming in and blood coming out, basically, and it goes around in the body. And you find this in different animals. So this is a very simple system in one way. But then you start to think about it, okay, what is this? Well, it can be more complicated if you think about the blood as a liquid. You have a flow in here. You have a flow in the heart. You have flow in the arteries, and you have some liquid walking around. And how to understand such a liquid? Then you can imagine a large computer simulation to understand the flow, the properties, and the issues about that. And then it becomes more complex. You can add layers of complexity here. If you take a microscope and you zoom in the heart, you find different fibers, different structures. You find cells, you find nuclei, you find borders between cells. It's not a random collection of atoms. There is structure in here. If you continue and look down into the actin-myosin cross bridges, you have a mechanical machinery enabling the contraction here, basically. This is very complex. And then if you go to more recent study, you can study different genes, how they control each other in different kinds of cells under different conditions during health and disease. And then the catalog of components is increased again, and you have a new layer of complexity here. So then we realize that the heart is, it's complex. It has many different scales in space, from small things to larger things, and also different dynamics in terms of times, from ion channels which are rapid to sort of more slow dynamics in a system. So then this looks like a complex system, typical kind of biology system. But then you feel that maybe we missed the point here. Well, what is the heart doing? It's pumping blood. Yes. Why does it pump blood? Well, that's not something you see when you look at the genes here. You don't have an answer to that question when you look at the different fibers. Why does it pump blood? It has to do with oxygen, of course, providing cells with oxygen and taking care of waste material. And these are important properties that a heart must sort of fulfill by pumping the blood, basically, and transporting this liquid. And you don't see these properties when you look in the system here. So then the question of system, what is a system, is very closely related to what kind of questions are we asking. What is a heart? Yes, it's a pump. But then we can ask different kinds of questions. What kind of genes controls different kinds of cells in the heart, for example? That's another kind of question. So we have to take care of being very careful about what are the kind of questions we're asking to what kind of systems. And that brings us into the issue, what are the relevant features, properties that we should think about when we think about the system? That depends on the question. So if we study disease, it may be very, very important to study misregulation of some particular genes. Or it might be anatomical sort of features which are erroneously in the very beginning when the child is born, basically. So it depends really on the question here. So it's just not a matter of sort of formulating a huge system of equation and then run the equations and then look at the equations and then think that we're done. We have to think about the questions. And this also addresses the issue that even if we can come up with a set of equations for a given system, which is a difficult problem by itself, because where do we find the data? Where do we find the parameters? How can we constrain such a system? And even if we do, it may well be, like with the oxygen transport, that we completely miss an important property or a function that the system is trying to fulfill, transporting oxygen, for example. Even if we have a very detailed description of a particular system, a particular structure in our bodies or an animal model, basically. And then even if we have that, there's also an issue, how do we find structures in this system which are relevant and persistent over time, the kind of thing of order that Schrödinger talked about. So for example, if you study fluid mechanics in physics, you can study wind, for example, or you can study liquids. And then when you study that, you find structures like these whirling structures in air, basically, that persist over time. That's a relevant structure that you want to understand. How is that propagated in this kind of system? Not necessarily dependent on every single molecule in the air. So you have the same kind of question here. How can we find a structure in these systems which are relevant for the kind of question we're asking? So these are examples of some of the hard problems we have when thinking about systems here. And we, me and others, have the belief that, like in physics, essentially, mathematics is useful and it's kind of necessary to understand these complex systems. We can have a verbal, intuitive reasoning about these systems, but eventually they become so complex with lots of different kinds of data and scales of separation in time and space, so we need a language to deal with that complexity. And that very language has turned out, for historical reasons, to be very efficient language in terms of mathematics, in terms of understanding physics. And we have no reason not to believe that this will be very, very important to understand life. Answering questions like Schrödinger asked, how can order emerge out of these atomic kind of compositions? So that is a bit background to the systems part. And we may think that now, 2015, this is really novel, exciting. It is. But people have, as I said, been thinking about this previously. So this is a very famous guy. There are many examples to take. This is one example, Norbert Wiener, very clever guy, He did his bachelor when he was 14 years old, one of these kind of geniuses basically. And he started to think seriously about what is systems and how are they regulated and how are they controlled, thinking about feedback system. It's not just a feed-forward system, but you have consequences of the actions of the system. And how can you create systems which are self-organized, persist over time, and even systems that can think and reasoning and act with the environment. So studying feedback systems, interconnected, complicated things, basically leading to very complicated mathematics, basically, and trying to understand, are there any rules, any principles here? So he invented the field cybernetics, it's called, very closely linked to artificial intelligence. And that was in the 30s, 40s, and 50s. He and others were working on this, basically, and trying to find some sort of laws and rules in this area. So he was an early pioneer. And these people were thinking about, as I said, building smart devices that can do things. And they were also thinking about different scales, different levels of organizations, from society to molecules in space and also time, and how to think about these systems. One issue these people had at the time was, of course, that the amount of data was fairly limited. So you end up working with your equations and thinking, trying to come up with ideas on how to deal with these kind of things. Because they had the intuition correctly, I think, that we must search for additional things in addition to atomic kind of properties here. But what is that, and how to deal with that? So then we're limited basically by our own imagination to come up with new ideas. So in biology, we have Drosophila, which is a very useful model system to understand system of molecules and components. And these guys need such a system as well. So what is their kind of Drosophila system? And another very genius guy, Stephen Wolfram, he did a PhD in physics when he was 22, and then he left academia and started his own kind of Mathematica company, basically, doing science in his own way. He came up, and this goes back to earlier work from Ulam and Neumann, people who really thought about the architecture of computing. A game, essentially, like a chess game. You have a board, basically, cellular automata. And every block in that square changes to the next time step. It can shift color from white to black, in essence, depending on how your neighbors are doing. So if you sit over here and your neighbors are smiling, it's very likely that you will smile in a couple of seconds again. So then you can devise games here, in the sense that you can have different rules for updating, depending on the neighbors. And then you have a pattern evolving over time. So this was a good Drosophila model in the sense of complexity. And von Neumann, before he died in cancer in the 50s, he thought about how can we build self-reproducing automata? How can we build systems that, for some rules and some time evolution, that creates a structure that persists over time? And then he could sort of mathematically prove necessary and at least sufficient conditions for that. And then people afterwards can find even smaller and smaller systems which could do that. But in a sense, it was not that satisfying because the complexity is completely wild. You have a very, very simple system. You have your chessboard, you have some rules, but the kind of patterns you can see are almost limitless, basically. So how can you find some structure in that? And the Stephen Wolfram's contribution in '84 was a paper in Nature, basically, showing that you can find structure for these rules and these systems that they can either be completely uninteresting, they die off, everything becomes black or white, there's nothing interesting coming there. There could be some periodic patterns, black, white, something like that. And it could be completely chaotic in a mathematical sense. You can't really predict what will happen in the next time step. But you also find a class of systems here with some rules that could be what we can call for self-organizing, that can produce structures, interesting structures persisting over time. So that was a major breakthrough in the Drosophila in the computer world, basically, finding some structure in here. as a toy system here. So I'm giving you these names and examples which you can later on look up basically and read more about that to have a sort of conceptual perspective where different ideas come from here, what people have been doing and thinking about these things. So what happened then? So this is really human history in 2 minutes. Then in the late 90s, we had an explosion, basically the emergence of internet. Coming from physics, and then around 95, 96, the web was sort of taking shape, basically. And what happened with this development was that the amount of data started to grow. So, approximately the same time as the decoding of the human genome, people realized that things like computers or websites, and you can look at traffic system, airports and that, is really organized in these kind of graphs that I mentioned earlier. You have nodes and you have connections. And they did not look completely random. So in math, we have this kind of random model of a graph. You take a coin and you flip, and then you say, yeah, let's connect A and B, and you flip again. I do not connect B and C, and you do that. You take a flipping coin, a random model, and then you have a graph, a mathematical object, and then you study that. And that has been the graph Drosophila for math people since long time back, basically. It goes back to Euler in the 18th century, essentially. But then things look very different in math world. But when people started to look at real systems here, how are computers connected with each other? How are websites connected to each other? How are actors in movies connected to each other? So you connect two actors, for example, with an edge, if they have been participating in the same movie, then you have a connection. And it turns out these systems are very different. The nodes are different. You have an actor, you have a computer, you have a website, or you have an airport. They show similar kind of structure, and it's not the kind of mathematical random coin flipping structure. It is the structure that you have some nodes which are very highly connected and then have more sparse connectivity. You have some kind of power law structure here. So this was really a revolution here 15 years ago, that networks in the real world really looks different from the mathematical kind of structure. And one of the pioneers, dropping a name again, is Laszlo Barabasi. So I recommend you to check his early papers and also later papers. So he was the one who, at least according to history, it was the first really to see this in the internet structure, basically. And he has been a pioneer the last 15 years of applying this kind of graph approach to understand very different systems, including systems in biology. So it's a rich source of publications here. And with the announcement of the human genome, we had even more data, and also more data coming after that in terms of technologies producing more data. So then we can study these different systems, including systems in biology with different kind of components here, like networks, and study their connectivities. There's a huge flood of papers using this kind of language to describe these kind of complicated systems. So that's very exciting. But this is the structure part. Recall that we had a structure and we also have dynamics. So at about the same time, people in applied math also looked at networks, these kind of complex networks, biology networks, electrical circuits, which are not like the random model, but has some structure. And they have a structure and they have a time dynamics and they evolve over time. So Stephen Strogatz wrote a very influential paper, 1999, where he had a very simple model, basically, like a ring of nodes, and they're connected nearby, basically. And then asking, what happens with this kind of system here if it evolves over time to certain rules, basically, depending on how tightly or how likely things are to be connected here. And it turns out that you have phenomena like in physics, you have a phase transition, everything becomes connected or is sparse. So then you have, again, a toy system to understand complex dynamics in these kind of networks. And it was very rich area. And so you should check up some of his papers, Stephen Strogatz. So that was another sort of development in parallel with the Barabasi development. And so what happens now, where we are now, is essentially that the list of parts is continuously growing. New data types, new technologies, more different kinds of nodes, which are somehow connected, related to each other at different levels of organization. We try to understand the structure in different organs, and we try to understand the dynamics. But then, as the example with the heart, we need to understand what are the kind of questions we're asking in each specific case. What are the relevant pieces? What are the relevant features to take into the equation, so to speak? And that is not the trivial thing. So these advances, as I said, really set the stage for revolutions. And with all this data, realization is that we need informatics and computer science. So you have the emergence of bioinformatics in the 90s, basically. But then when to understand biology, it's not just enough with bioinformatics to process these kind of data. We need to put things together. So 10, 15 years ago, a lot of sort of high-profile reviews on systems biology started to appear, people talking about the necessity of taking and integrating the data. And I think they were completely right in pointing out that very need, but how to do that? That's something we're still working on, basically. So to understand an organism, it's not enough to chop it up into pieces and measure everything from the bottom to the whole system, basically, like the heart example. We need tools for how do we pull this together. How does such a system work? How does it produce order over time? Like Schrödinger's second question, like we saw also in the heart example. And the revolution in terms of data continues. So it's not only the molecular revolution. Today, the last 5-8 years, with the emergence of the cell phones, smartphones, and also different kinds of sensors, we can add on the data flow. So characterizing the phenotype, if you so like, how we live, how we move, how we do, in combination with the molecular revolution. So the data space is kind of increasing not only in size, but also in diversity in terms of different kinds of data. And the question is, again, how do we think about these different data in combination for different kind of questions? How do we integrate this into something that tells us something, what is health and what is disease, and can we detect disease early on? So there's enormous kind of promise in here. Many people think that in not far to future, we will have the vision that when a baby is born, more or less, and in the whole lifespan, we will have a data cloud. Of course, we have our DNA, the complete sequence, and we have lots of other data types. We have transcripts, we will have proteins, we'll have images recorded over time, in addition to sensor information from different kinds. And you can add in also social information. So it's very realistic, I think, to see that within a couple of years, 5-10 years, we will be surrounded with these data clouds on individual level. And that's an opportunity and also a challenge for medicine. And I do think systems medicine will be one very, very important approach in trying to address these kind of opportunities and challenges. And of course, all of this speaks for that we really need powerful algorithms to deal with this kind of data. Preferentially in a data-driven way, because we cannot really hand-tune algorithms for every specific case. And there are very exciting developments over the last decades in terms of faster computers, more powerful algorithms. We can process more and more complex data and do computations of that. And also, we have algorithms that start to become really smart and data-driven that can look at different kinds of data and try to extract a structure from this data. So understanding this space in combination with the data explosion and having a grasp on the kind of questions we're asking in the context of understanding diseases will be key and is key currently. So what will happen now in the summer school essentially for 1 week? I would say this. In essence, you will be exposed basically from different speakers having different perspectives on how to use this in different combinations, understanding different diseases, presenting their kind of success stories, but also discussing hard challenges in these kind of efforts basically. So we hope that we have chosen some good examples here, and as always, failures and challenges are really instructive because that points to us what is it that we need to develop further, what do we don't understand, do we have innovative concepts enough and methods to deal with this and try with that. So that's for the historical perspective. So I will spend last 5-10 minutes on some sort of more advice. So that's about the schedule. And who will you meet here? So I'm very happy to have a team of people here helping out with the summer school. It has been lots of planning also with the support of the Casym community, the Coordinated Support Action for Systems Medicine, and FEBS has been helping also, financial support. The one who has been really coordinating this, and you have been in contact with her, is really Rafaela, sitting there. So she is the master, the spider in the web here, and also with the help of Pernilla to making this really happening with all what it takes. And also today we have 2 students here. We have Sudabe and Sanjay working with our team here basically. You will talk more about your research projects during the poster session and that later. So they will help up. They will help basically Rafael and Pernilla with practical things during this week, essentially. So what about the summer school? Well, what kind of things should I say? The second paper you have here is a list of participants. And as you can see, you're doing different things, you have a bit different background. So it's very clear. Interact with your fellow students. Talk with each other. What are you doing? Why are you doing this? Yes, my advisor told me to do this. But what's the rationale? What could it lead to? And get to know each other, because in the future, you travel to different places, you go to meetings, you will hook up again, and it's always good to expand your sort of professional network and know who to ask about what, basically. I think because this science, to understand these kind of complicated things, requires many different mindsets. So it's very, very important to be able to interact and collaborate with other researchers. And not only other researchers, but people with different kinds of training than you have. So if you're a math guy, you really need to work with people who understand biology and vice versa. And also, secondly, take the opportunity now to interact and ask the faculty people who come here. They give the talk, they stay 1 or 2 nights, a bit different depending on different speakers, basically. Ask questions during the talks, use the coffee breaks, call them in the morning, 2 a.m., not really, but use the evenings also. Really take the opportunity when you have these people here. Ask them tough questions. Tonight we have a poster session, and the idea is that this will be a quick exposure of what you are doing essentially in your projects back home. And also the posters will hang here for the week, basically, so you can sort of go back and talk about your stuff, basically. So we have 3 different rooms. We have this one, and then we have 2 other rooms here where we can hang posters. And we hang posters in 2 rooms there, so we can sort of mingle around and look at the posters. And tomorrow we have a social, bit longer dinner, interact and meet and talk, basically. And as you've seen, this is a nice place. There are some activity possibilities here where you can be outdoor doing things, or you can also go to the gym, or you can do some swimming, which is nice. So do that whenever you have free time. But you will be working in small groups. So by tomorrow, you will have an introduction for different tutorials, examples, demonstrations, and you will have small projects in small groups, and you will sort of discuss your achievements on Friday morning. Now, how is that going to work? And then you have 3 more names here. So you can see David sitting in the back there, and also Francesco here. And Gordon will arrive on Wednesday from a meeting, basically. So David is trained in math, also medicine. Francesco has a background in human genetics, biology, and bioinformatics, basically. And Gordon is trained in physics, again illustrating the kind of different mindsets. So they will lead these efforts in having a couple of demonstrations and also project work and assignment of these kind of small groups. So these are important people. And we really expect you to work specifically hard on, say, Wednesday and Thursday in particular on these kind of projects in the evening. Now, that's about the practical part in terms of who you are and what to interact. And then I want to highlight 3 kind of concepts that you can think about and ask questions during lectures, in coffee breaks, and whenever you have the opportunity. Ask your fellow student here, ask faculty. You will hear a lot about networks. People talk about networks. What are they talking about? What kind of network structure are they really talking about? What are the edges? Do they have any direction or no direction? And how is that basically constructed in these different examples? And what's the semantics? What does these edges mean? Well, if you take a simple example here, you realize immediately that these edges could mean different things. It could mean that a protein is controlling a gene, or a gene is effectively controlling another gene via a chain of events, or it could be some metabolic interactions, or maybe a collapsed version, or it could be interactions between cells. So always ask yourself, what kind of graph are we dealing with here? What does the graph mean? What are the edges annotating, so to say? Where does the data come from? These are always important things to keep in mind to understand and have a more precision. So networks first. Second one, which we've been alluding to, is like the heart example, coarse graining, like the physicist would say, scales, separation in space and time. When we try to understand a system, what is relevant level of coarse graining? Are we understanding a single gene or some large kind of network or something in between, the system complexity? What are the kind of relevant detail in terms of representing the system and understanding the system? Do we need some kind of very stochastic simulation? Or it's enough with kind of heuristic graph approach or something in between here? What is the relation between the question I have or the questions and the kind of data I have available? What are the relevant tools and levels of coarse grading to look at the system here? And the same with information here and dynamics over time here. So these are things to keep in mind. So studying the heart as a pump, for example, is it relevant to know exactly what this kind of gene is doing or not? The third one is the difference between statistics and mechanics. In many cases, it's enough to make a decision. You find basically in data space a separation, a boundary. So in machine learning, you have these kind of algorithms that find efficiently these kind of boundaries. This is white, this is black. We don't care about why, but we have an efficient procedure for separating these kind of things. And that's good enough. When we can make a decision. Should you have this treatment? Yes or no? Based on experience on these other patients, yes, you should have it. We don't understand exactly why, but statistically, it's the best thing we can do. And in many cases, this is good enough. And when we do statistics, we have to be mindful about what kind of complex statistical model do we have. We may have some very complicated data, and we make a statistical model here that fits perfectly with available data, but we overfit the data to such a degree that it doesn't generalize to a new case. Or the model might be, or the wrong class, it might be a poor representative. How can we find a model that captures relevant amount of complexity here without overfitting and have some kind of predictive value when we have new samples? So this is important to think about when we're dealing with statistical models. But statistical models never really open the box in the sense of how do these systems work. And when we ask this kind of question, how do these systems work, we most likely think about causality. What would happen if we increase the activity of that gene? What happens to the cell? Or if that pathway of that cell is blocked, or we interfere with the communication between the cells in this particular tissue. If we do A, what will happen then? And then we essentially asking causal kind of questions. And when we just have a graph with no dynamics, no rules basically, it's very difficult to predict what will happen if I move these nodes and edges. In many cases, we need a mechanistic model about the mechanism basically, so we can really see through the equations what happens if we interfere with the system. And then we come back to really hard causal kind of questions, which we can deal with when we have fairly simple questions like model organisms like a Drosophila or an E. coli or potentially a yeast. But when we deal with human diseases, the issue of causality is much more complex. So my final slide here before the coffee break is basically asking you this. So you should view this as an opportunity. We're here for a couple of days basically. Your fellow students, your faculty, and really make most use of your time. Ask tough questions and interact with each other and check the things that speakers talk about. So that's all for that introduction. Any kind of questions? And speakers will introduce themselves, and some speakers may prefer to have questions during the talk, and some prefer maybe to have them after the talk, and they are free to decide themselves. Okay, let's go for some coffee.